Hello everyone. So today we are going to talk about a man which you are seeing him on screen. So can you just tell me who is this man? Yes, he is a very famous man. Absolutely right. So his name is Gregor Johann Mendel. Yes. And who is he actually? Yes, absolutely right. He is the father of genetics. Now, as we have studied in our previous class, that he is famous for giving all those three laws, okay, which governs the whole genetics now. Okay, so law of dominance, law of independent assortment, and law of segregation. Today, in this class, we are going to talk about the different ratios in a dihybrid cross. Right. So for that we have to move into this particular plant. Okay. So yes, absolutely right. This is the same plant which Mendel has used, and this is the pea plant, the Pisum sativum that we normally call it. Okay. So here you can see there are two types of seeds in this pea plant. One is yellow, right, which you can see here, yellow, and round. Right. And what is this? This is phenotype of this particular seed. Right, everyone? And because it got two characteristics. Now, Mendel has selected these two characteristics, and that is why it's called as a dihybrid cross. Fantastic. Okay. Now, this plant, on the other hand, the seed of this plant is green and wrinkled also, right? Not round, but very much wrinkled. Okay, so this is the phenotype of this particular seed, right? So there were two types of seed that Mendel has noticed. Okay, now what he did at that time is that he crossed both these, two different variants, we can say, right? And as we know, we have understood that these are the phenotypes: yellow and round, green and wrinkled. What can be the genotype here? What can be the genotype here? So if it is yellow and round, the genotype here will be capital R, absolutely right, and capital Y, right? What could be the genotype here? Because it was, at that time, as we have studied in earlier class, that this was a dominant characteristic. We have written it into caps, right? Now, as green and wrinkled is a recessive characteristic, we are going to write it into small letters. So small r, and small y. Absolutely right. Okay, so we have all understood it. Now, the thing is that he crossed both these, variants, and in F1 generation, so this is the F1 generation. In F1 generation, he got all the seeds. Okay, he got all the seeds, which were surprisingly, all of them were yellow and round. All of them. Okay, so this was pretty much different from monohybrid cross, right? But he didn't stop here. He went for another self-crossing of this particular plant only. Okay, so we call self-crossing. Why self-crossing? Because he uses the same plant, okay, and did a self-pollination, right? Okay, so that the particular allele will remain intact. That means if it is capital R, Y, it will cross. If it is small r, small y, it will get crossed. Fine. So what are the different, hybrids, different gametes we can get? Okay, is from this cross is, if we'll try to write it here, is R, Y, okay, capital R, small y, right, small r, capital Y, and small r, small y. Why is that? Because this plant was R, and Y. Fine, because it get crossed with small r, and Y, capital R, small r, and capital Y, small y. Okay, and that is why you will get these gametes for F2 generation, right? So these were the gametes for F2 generation. Now Mendel crossed it simply. Now after Mendel, there was a scientist called Punnett. Fine, Punnett. So Punnett has actually expressed this thing into a beautiful square, okay? And today we are going to use that simple square only, okay? So here is that square, right? All of you can see it? Yes, absolutely. Thank you. Okay, so what we have done here is that we have taken those gametes, one from, you can say, male, okay, and another one, you know, this one is male and this one is female, right? Same gamete because he has self-crossed it. And this is the Punnett square that you are seeing here, right? Now, what is happening? What do we want right now here is genotypic ratio and phenotypic ratio, right? That what we wanted right now here. So first we'll see the phenotypic ratio of a dihybrid cross, right? Now what you have to do, you just have to cross, okay? Write it down. What should be there in the first cell? Yes, tell me what should be there. Absolutely right. Capital R, capital R, capital Y, capital Y. Absolutely right, because both the gametes are having capital R and capital Y, right? That you can see here also. Okay, so like that I have, so here you can see capital R, capital Y. So like that I have selected all those cells which have at least one R and one capital Y. So you can see here that every cell, which I have named here, okay, according to their cell position, they have at least one single capital R and one single capital Y. Why I have selected that? Because if there is only one capital R, that particular phenotype will be round. If it is capital Y, that particular phenotype will be yellow. So all these plant which have these genotypes will be round and yellow. That you want to see here, and we can see here like this. Yes, so every single seed here is round and yellow, right? How many of them? Can you count? Yes, how many of them? Absolutely right. It is 9. Okay, so we are writing phenotypic ratio. Fine, we are writing phenotypic ratio, so it is 9 here. No doubt to anyone. So again, if you can see, it's making a very beautiful triangle, right? It's making a very beautiful triangle, right? Exactly. Now we'll see another one. Now this is round and yellow. What other things he got at that time? Here. Now I have selected at least one capital R and small y. So all these three places, so it has at least one capital R and one small y. Okay, what should be the genotype here? Yes, round but not yellow, it's green because small y is there, right? So what should be the ratio here? So I'll write it with the green only. What should be the ratio here? Okay, so it's 9:3. And again you can see it's making another triangle. Can you see that? Yeah, exactly. Okay, let's see another one. Now what I have selected another another 3 cells which have at least one capital Y and small r. So here you can see at least one capital Y and small r. Okay, so what should be the phenotype here? Can we see? Can you tell me? Very good, absolutely right. So it is yellow but wrinkled because capital Y was there, small r gave it a wrinkled form, right? And you tell me what should be the ratio here? Absolutely right, it's again 3. And again if you'll see here, it's yellow. Okay, so I'll take another pen to actually so it's again a triangle, right? Just to give you a a pattern like thing. Okay, so we have actually gone through 3 triangles. Now what a very single cell left here, okay, that is the last cell that you can see here, is this one, small r, small r and small y, and absolutely without any guesses the phenotype will be, yes, and it is showing here green and wrinkled. Very well, very well. So last but not the least, the last ratio is 1, and this is the phenotypic ratio of a particular or a typical dihybrid cross. Fine. So we are going to remember this one, and we'll try to understand the genotypic ratio here. Fine. All right. So I'm just moving down to my particular. Okay. So we'll try to understand this thing through another round of Punnett square. Fine. All right. So we'll try to do it with another round of Punnett square. So this is your Punnett square, right? Can you see it? And yes, on the very first one, it's again written capital R, capital Y, capital Y. So 2R, 2 capital R, 2 capital Y. Okay. So we'll try to again do here the genotypic ratio. Fine. All right. So let's write the genotypic ratio. So I'll try to write the genotypic ratio at the bottom, okay, with the red pen. Okay. So again, I'm choosing all those cells which have both capital R and capital Y. Okay. So now I know that there is only one cell in which it is possible. Okay. So there is only one cell. You can also see that there is only one cell in which it is possible. Okay. Right. Then I'll see you will see another magic here. Now there is only two cells which have both the capital R and one capital Y and small y. So you can see here, both are exactly same, a diagonal relationship. So two cells having the same genotype, that is why the ratio is 2. Right? Now, see again. Again, two genotype, okay, or two cells which have same genotype, or two seeds which have same genotype. Okay, fine. Here you can see, same, exactly same. So what we're going to write it here? 2 again. Fine, all right. Let's move again. Now, yes, there are 4 genotypes, okay, which are exactly same. Capital R, capital Y, small y. Okay, all right. So what should we write here? Write here as 4. Okay, right. Now next again. So it will keep on repeating, like now you can see here. It's a diagonal relationship. All of you, it's a diagonal relationship. Another diagonal. So here you can see, right? We'll move further. Okay. So what I'll do, I'll try to write it here again for all of you. So I'll write it at the topmost position. Okay. So till here, what we have understood, that there is 1 is to 2 is to 2 is to 4 is to 2 again is to 2 again, right? Okay, so this is 2 again, a diagonal relationship, definitely. Now next we'll move again. So again 1. So there is only one which have this particular genotype, right? But yes, this was having 1 and this was having 1, but still there are 2 left, this one and this one. They are also 1 and 1 only. There is one only single genotype here and one single genotype here. So that is why I'm writing 1 here, and that is why I'm writing 1 here. So what you're seeing here at last, when you'll see here, so these diagonals have only a single genotype. There is no pairing here. On the other hand, if you'll see this green color, definitely it is same, right? If you see this particular genotype, okay, this particular genotype, it is same. If you say, this diagonal, it's same. If you see this diagonal, is same. This one also, right? And that is why there is only one is to 1 is to 2 is to 2 is to 4 is to 2 is to 2 is to 1 is to 1, right? Again, simple, just magic. Or if you can just draw, a mirror image on 4, so it's just like this one is mirror image of this one. Isn't it amazing, right? So this what exactly the genotypic and phenotypic ratio of Mendel's dihybrid cross. I hope so you have understood this particular topic. We'll meet again next time with such another topic. Goodbye all of you.